Okay, so record is on, and like I said, we're going to do membrane transport practice questions and talk about metabolism, chapter 4 information, and go through some of these lovely blank diagrams. So hopefully if you planned on printing that out, you went ahead and did that. So I have my chat box open. So if you need to ask a question, you can turn your microphone on, or you could type me a question in the chat box. But we're just going to go through some practice questions here. And I'll pause in between. And I'm trying to let it to do a slideshow. There we go. So I'll pause in between, and I want you to see if you can think of the answer before I give it to you. So number one, which of the following is nonpolar? So which of these wouldn't have a charge to it or a partial charge distribution? Fatty acid tails, water channels, water, or phospholipid heads. So this is about the cell membrane, and remember it's made out of phospholipids. And so we've got a polar section and a nonpolar section. And do you remember which of these is nonpolar? It is the fatty acid tails. And I need to see if I can make my little, there we go.
Is this,
might be able to highlight.
Is this
Oh,
in the packet with the diagrams, or is this, where are these questions from?
Pin, haha. Okay. So there we go. Now you see how great I draw with a mouse. Okay, number two, the term fluid mosaic model refers to an experiment that shows the function of the cell membrane, the dynamic flow of proteins and lipids within the membrane's ability to communicate with the fluid portions of the cell, or the cell's ability to communicate with the fluid portions of the membrane. So it's tempting to think it has something to do with communication, because the second, the third, and the fourth options both do communication. But if you go back and you look at the PowerPoint on the second half of chapter 3, the fluid mosaic model is referring to the structure of the plasma membrane, and that essentially, because the lipids and the proteins aren't bound to each other within the membrane, they just have nonpolar and polar interactions. The phospholipids are able to, move around, and so are the proteins. And so the fluid mosaic model refers to this flow of proteins and lipids within the membrane. And so they're not bound, and so they can move around, and that's what fluid means. And mosaic actually means that the membrane all around the outside of a particular cell may not be the same on all sides. And I think you saw a little bit of that when you were looking at the slide images during lab module 3.
Where are these questions from?
Okay, for number 3, an organism living at the equator has more saturated phospholipids in its cell membranes than an organism living at the South Pole. Why? And this is getting at remembering that if a fatty acid tail is unsaturated, then there are double bonds between at least 2 carbons in one of the tails, and those double bonds means that the chain isn't saturated with hydrogen, and therefore that chain is kinked. And when the fatty acid chain or fatty acid tail is kinked, it pushes further apart from the phospholipid next to it. And the more kinks, the more double carbon bonds there are, the more unsaturated the fatty acid tails are, the more unsaturated the phospholipids are. And so that's, if they're kinked, they're unsaturated, and if they're saturated, they're straighter and they fit closer together. So why would something at the equator need to have more saturated phospholipids? And if you think about it, it's hotter, and the hotter something is, the more readily it melts. And phospholipids are lipids, so the warmer it is, the more fluid a membrane becomes, and we need to have more saturated or straighter fatty acid tails in order to keep the membranes more tightly packed and to allow the cell membrane to control what crosses them and to not be leaky. So which of these matches? So at the equator, having more saturated phospholipids. So here, in cold climates, more unsaturated fats with kinked tails are needed to maintain the fluid. So unsaturated does go with kinked tails. So that one's okay so far. And by pushing them further apart, they would maintain fluidity. In cold climates, more saturated fats do not go with kinked tails because the more saturated it is, the straighter it would be. And so we know that we don't like this. Give me a second. I'm pausing my share.
He's
Now
not
we're
hearing
recording
anybody.
again after a pause. My 1-year-old was yelling in the kitchen. Okay, so number 3, let's take a look here. We've got warm climates, more unsaturated fats with kinked tails. Okay, so let's look at it. Does unsaturated go with kinking? If it's unsaturated, it's going to be straight or unkinked. So we know that that one doesn't work. Now let's look at the bottom one. In warm climates, more saturated fats with kinked tails are needed to maintain the fluidity. Now, do you remember what I said at the beginning? Saturated fats with kinked tails. All right. So if we're saturated, are we kinked or unkinked?
Unkinked.
Anybody
Kinked.
remember how that goes? If
Kinked,
you're
kinked,
saturated, then
we
you
need
have
to go in the
maximum
chat room and let her
hydrogens
know she's not hearing us.
or no double bonded, and so you would be unkinked. And so
I wrote
that one doesn't make sense either. So we want unsaturated fats with kinked tails to maintain fluidity in cold climates. So that matches the South Pole. And then conversely, an equator, Lindsay asked where are the questions from. They're awesome. Actually, I can't hear anybody.
Yeah,
So
I know.
I'm not muted. Let's go. All right, so just wrapping this up with number 3, it hopefully makes sense that unsaturated fats, because they're unsaturated, they have double bonds because they are missing some hydrogens, and so some of the carbons are double bonded, so the tails will be kinked. The more unsaturated, the more kinked they are. I think she'll survive. Take her upstairs and play. And so that helps keep the cell membrane from being packed too tightly, and it maintains fluidity at cold temperatures. Now, at the equator, we don't want to get too leaky, so we need more saturated, unkinked, straighter phospholipids so that the membrane can stay together. And it's still fluid, it's just packed tighter together because the higher temperatures keep those phospholipids further apart. All right, so number 4, the phospholipid tails of the membrane face one another because, and you may remember me drawing and then referring to an image online last week and talking about how the tails are non-polar. So they're the hydrocarbon chains, and so non-polar is the same as hydrophobic. And so we want to look here at hydrophobic and hydrophobic, and that, you know, of course, gets rid of either of these two options, right? And then does hydrophobic mean attracted to water or repelled by water? And so you have to basically match these. And so this becomes our answer, that the phospholipid tails are hydrophobic and they're repelled by water, and so they turn toward each other to escape the water that's the interior of the cell membrane and outside in the extracellular fluid. And they didn't shut the door upstairs. One more time. Okay, so number 5, which is not a function of cell membrane protein, cell membrane proteins. So we're squeezed out a little bit here on the edge. It should say plasma membrane all the way over here. So you would need to go back to the PowerPoint that was dealing with membrane function. And remember, the phospholipid bilayer is what isolates the inside from the outside. So it isolates the contents. And the proteins are what do the job of controlling what goes through. So it would do this. So it is yes here. Remember, things can pass through. So this is a yes. And it does, we have proteins that are embedded that have special tags on them, like carbohydrate chains. So this last one is a yes, and I left the third one. Remember what I said, what is it that isolates the inside of the cell from the outside? That is the bilayer, that's the lipids. Oh, that's really great P right there. Okay, so this is not a job of the protein, and so that makes this the correct answer. Okay, number 6, which is not a form of active transport. Okay, so remember, active transport requires energy, okay? And so if something requires energy or ATP, then it's active transport. So which one of these does not require energy? And if you remember, pinocytosis is cell drinking, and that means the membrane has to pinch off and make a vesicle. So that is active transport. Receptor mediated transport, that means there's going to be a protein embedded in the lipid bilayer that has something bind to it, and then there's a signal pathway that's followed, so that's active transport. And phagocytosis is when a cell engulfs a whole nother object, whether it's food or it's something that's going to then fight from the immune system, so that's active transport. And osmosis is the diffusion of water. I bet after a couple of weeks of drawing like this, I might get really good. This is where I wish I had a touch screen. So the diffusion of water going down its concentration gradient. So I'm going to put the little brackets around water showing its concentration gradient. And so since it's water moving from towards solute, or from high free water availability to low free water availability, then it would be passive transport. And so it matches being not active transport. And so I would choose osmosis. Okay, number 7, which cell has the highest surface area to volume ratio? This is something in the PowerPoint about cell structure. And so units, instead of giving you an actual distance, you know, we could use centimeters, we could use meters, we could use millimeters, it doesn't matter. Let's just use them relative to each other. So here's a cell, we'll call that 10. Let's double that and call that 20. That's really hideous. Here's the third one that we're calling 50. And then here's the fourth one that we're calling 100. And so the one that has the shortest distance across is the one where the radius, which would be half of the distance, would be 10, and the full diameter would be 20. And as you increase, both in size, volume goes up and surface area goes up. But as cells increase in size, volume increases faster than surface area does. And so the one with the best surface area to volume ratio is this first one, because as we go up, yes, all of them have a higher surface area, but because this internal volume is so big, the surface area to volume ratio is the worst in a bigger cell. So this one with the 10 units has the best surface area to volume ratio. And we could go into, you know, how to calculate surface area versus how to calculate diameter of a sphere, but we'll just, if you want to go into that, go back to the PowerPoint that's
So if,
on
if
that.
there was another choice that said a cell with a radius of 5 units, would that have a
Yeah,
better?
so,
okay.
so small, smaller cells. So what is limiting cell size? That's really what that is getting at. Smaller cells, the smaller you are, the better your surface area to volume ratio. And so this gets us to think a little bit about, and I'm going to put prokaryote and just put P versus eukaryote. And why is it that prokaryotes are little and eukaryotes are bigger cells? What is inside a eukaryote that allows it to maximize surface area to volume ratio and be able to be bigger but still be efficient? And that's
Organelles?
because they have organelles that have interior membrane surface area, and so they can have a bigger volume but still have sufficient surface area because their organelles compartmentalize functions and they maximize surface area for things to be done. For work actually gets done by proteins across a membrane because of creating concentration gradients, but there's still going to be a maximum cell size. We don't have cells that are really visible to the human eye because we still have to get from the outside to the center in a meaningful amount of time. So if you're thinking about like a cell needs oxygen, right? And the oxygen has to go in, and what does it need to get rid of? Oh, that's a really bad O. That's supposed to be CO2 for carbon dioxide. Okay, so and a cell creates waste. So let's talk about, you know, something like urea, where it's nitrogen waste, that if it doesn't get rid of it, it's poisonous. And we need to take nutrients besides, you know, oxygen in. We need to take nutrients in like glucose for metabolism. I'm really writing about like my son does right now with how neat my writing is with the mouse. So we need to take nutrients in, and if the nucleus is in here in the middle, right, it needs to get oxygen in order to function. And so there's a distance, there's a maximum distance you could move and still have nutrients and materials coming in and waste being removed for that cell to function. So what happens to a eukaryotic cell once it reaches its maximum surface area to volume ratio before it becomes inefficient? Well, then it would do what?
Divide.
Yep, it would divide. It would go, and so that's what I'm showing here. It would divide. And a prokaryote would go through asexual reproduction. It would split and clone itself, okay? So eukaryotic cells are bigger because they have organelles to compartmentalize functions and maximize the surface area to volume ratio. Okay, so the next one, a semi-permeable membrane separates two solutions, permitting water but not larger molecules to pass. Okay, so let's draw this here, our little container. Okay, and we have a semi-permeable membrane, so I'm going to draw it as a dashed line down the center. Isn't that lovely? Okay, and we'll say this is side A and this is side B, okay? And side A is 20%, and we're just of a sugar solution, and we're not getting really specific about which one, and side B is 40%, okay? Whatever the sugar is, it lets water, but not the larger molecules pass. So water is here on both sides. And the water can move both directions, right? But the sugar can't move. So remember, water follows solute, right? So the water would have more movement. And so I'll draw a bigger arrow this way. We're going to have water moving this way. But when we look at this at equilibrium, what's going to happen? So initially, and this isn't a cell at all, so we're just talking about something being tonic, not osmotic. So initially, A is hypotonic to B, but when we reach equilibrium, what will have happened here? If there's enough room for the water to move, then enough water will move so that the solute concentrations will match. And so when we reach equilibrium, we'll be isotonic. So my daughter's here next to me wearing a fire helmet, okay? That's really hilarious.
Safety
Okay,
first.
that's right. You know, one of the plastic kinds you get when you go to the fire station. So initially A is hypotonic to B, and B would therefore be hypertonic to A, right? But when we get to equilibrium, all the water has followed the solute, and then we would be isotonic. So remember, this is equilibrium. So don't get so caught up in what you see initially that you would accidentally answer the question incorrectly. Make sure you look for that.
I have a
Okay.
quick question.
Yes.
So if A is hypotonic to B, but so if we were talking about a cell, and a cell would then take in more water, at what point do we know if it's going to reach equilibrium or if it's going to burst?
Once you've run the experiment.
Okay. Okay. So like if we had a hypothetical question like that, then there wouldn't.
Well, let's see what's next. Okay. Well, I wanted some white space is really what I wanted. So just to continue your question, so if I draw a beaker here and I draw my little model cell made with the dialysis tubing and with like string or something tying it off, and if I have solutes in here and the membrane is permeable to water only, and I only have a few solutes out here, then I know that my water is going to enter. And so at some point, you're either going to reach equilibrium or the cell is going to lyse. So if it's a model cell, the way you would do it is you would, if you were running this experiment and you didn't want to lyse it, you would roll it out, and then before you tied it off, you would actually kind of create this tube that the liquid is in here, and then you would have this section here that was sort of flat, and then you would, and without air bubbles, and you would tie it off so that it would have room for the water to enter without exploding. Yeah, in our lab that we do on this, we say no sausages, and students are like, what? Well, it's because if you make a tight little sausage roll with no room for water to enter, then you end up with a leaky cell and bad data. If it's a regular cell with a nucleus, then if it has lots of concentrated solutes in here, then water's going to enter, and it basically has to then be able to pump the water out. So think about something that lives in freshwater aquatic organisms are going to be hyperosmotic with respect to their outside. And so a freshwater organism would always need to be doing what? It's going to have to pump water out. in order to survive. And a saltwater organism is going to have to maintain homeostasis by figuring out how to get enough water from the salt water that it's processing without accumulating too much salt. So it has to figure out maybe how to get rid of salt or how to move water across. And so things like salmon, that's been part of their life in freshwater and part of their life in saltwater, are crazy adapted because they can deal with osmosis where water's following solute in freshwater, and then they can conversely figure it out in seawater. So another good reason to be a marine biologist, all of that stuff, right? Okay.
Thanks for
Ready
explaining that. That makes sense.
You're welcome. Ready for number 9?
Yeah.
Okay, which membrane process requires molecules to bind to a membrane protein? Okay, so when we're talking about molecules binding, then we're thinking about maybe active transport and a change in shape. So does water moving through channels, specifically aquaporins, need a change of shape? It doesn't. This is just going to be osmosis, where water is moving through the specific protein channels, and that's going to be facilitated transport, which is passive diffusion. And we have a visitor. Okay, then oxygen diffusion through the membrane. Well, oxygen again is going to be following its concentration gradient, so this is also passive. And look at this one. We talked about this before, receptor mediated. Well, if something is receptor mediated, then here's our membrane, here's a protein embedded in the membrane, and a little molecule has to come and bind. And then in scenario 2, we'll just show this with a changed shape. And now that it's bound, and there's going to be a shape change here based on its binding, and it's going to change something that happens inside the cell. You could read chapters about cell signaling pathways and about how molecules like hormones come and bind to proteins, and that causes a cascaded reaction through the cytoskeleton to the nucleus, and then that turns genes on and off, which tells the cell what kinds of proteins it should or shouldn't be making at any point in time. So this one, receptor-mediated transport, is going to require molecules to bind. And this one, lipid movement across the membranes, well, it doesn't specifically require binding. This question isn't asking about active versus passive transport, and that would sort of really depend on size, but something that was small enough could, in theory, use a channel protein. And even if it couldn't, it may not require binding, so to speak, on the outside. It just might be a different kind of transport altogether. Okay, so that was number 9. Number 10, facilitated diffusion uses what kind of protein? So remember the diagram that I went over that explained simple diffusion is just straight through the bilayer, and then we have facilitated diffusion, which is also passive transport, and it uses either channel or carrier. Right, so much nicer on the board. And so which of these do you see here? Oh, well, you would recognize carrier, okay? And it might help to go back to chapter 2 on biological molecules and refresh yourself on all the different kinds of proteins there are and what their functions are. Because proteins are really diverse and they do all the work of the cell. Okay, number 11. Oh, here we go. This kind of gets at a little bit at what we were talking about before. So the salt concentration of the fluid surrounding a cell is more concentrated than the fluid inside the cell. So if we have our little cell here, and then we're thinking about the extracellular matrix that it's in, you know, so here's some cells. If I had drawn these better, I could have called these simple cuboidal cells, right? Here, we'll do one square just for kicks. Okay, so the salt concentration of the fluid surrounding the cell is more concentrated. So that means I get to make a whole bunch of little clicks here. And then inside my cells, we'll just do a little bit, okay? So outside would be, do you remember what this one is?
Hyper
Yeah, hyper what?
osmotic?
It's outside the cell, so the external environment is hypertonic, and inside the cell, it is hypo what? Hypoosmotic.
And is that really the distinction? So hypertonic is speaking specifically to what's in the cell? I just could never quite understand that
So
distinction.
osmotic is the cell with respect to its environment. Tonic is the environment with respect to the cell. And if we're not talking about cells and we're talking about like two sides of a simulated membrane in a container, then we would use tonic. If we're talking about a model cell in a fluid, then we would refer to the cell with blank osmotic and then to the solution as blank tonic. And so hypo means less and hyper means more. So the salt concentration of the fluid surrounding a cell is more concentrated. So we know more means hyper, and because it's outside the cell, it's hypertonic, and the fluid inside the cell is less hypo, and because it's in the cell, it's osmotic. There's some more tricky stuff to do with osmolarity than what we're getting into here, but as far as hypo and hypo go, that really works, okay? So think about this here. Which way is the water going to go? Water follows solute. So water will leave. Okay? So the water is leaving the cells. And so what happens when water leaves the cells? They are going to
Crenate.
well, they're going to shrink. And then if it goes too far, then they'll crenate, right? Just like so we could write yes, they will eventually crenate. If you took a blood sample and put it on a slide and you added fresh water, you would see the cells swell and then they lyse, and then actually after a few minutes you can't find any cells on the slide at all because they've all exploded. And if you put salt solution around the cells, you can see them shrivel and get all spiky, and then they turn into like tiny little fragments. It's pretty cool, actually. All right, so number 12, this is about cell junctions. So there were a couple of slides that we looked at in the first PowerPoint on chapter 3, and these were kind of toward the end. So if you remember what tight junctions make things waterproof. So it's a whole bunch of proteins that are going to bind, you know, hundreds or thousands of neighboring cells together, you know, to maybe create a tube, right? So that things that go through this tube, you do not want them leaving. So you need all your cells that are making this tube to be what?
Tight.
Tightly bound together with tight junctions. Okay, and then, so I pretty much gave it away. So to prevent fluids from leaking across that layer, we want tight junctions, okay? And desmosomes, those are basically proteins that bolt cells together, okay? And Gap junctions are when we talk about animal cells. For communication. Oh, I can do cursive so much better. But it's even harder to read. And plasmodesmata are tubes, protein tubes that are in plant cells that are for communication. Okay?
Oh, yeah.
Okay. So number 13, active transport. Ooh, that was a bad underline. That's hilarious. The energy source that moves a molecule against its concentration gradient. What's the ATP? I just gave it away. The molecule that moves things is ATP. Now, these are energy carrying molecules, but their job is basically to carry energy to help make ATP. And glucose has energy, but we don't move a molecule against its gradient with glucose because a glucose can make about 38 ATP and it's just too big. We split it, and when we first split it, we make a couple of ATP, and that's what we're going to get into next. All right, number 14, I think there are 2 more slides here. Number 14, do you remember how animal cells communicate with each other? What did I draw on the last slide?
Gap junctions.
Gap junctions, that's right, okay. And then, your favorite. Which of the following, which of the transport processes in the figure below results in the movement of molecules from a high to low concentration? Okay, so that means which of these is passive transport, moving from high to low? Okay, so simple diffusion, high to low. Facilitated diffusion with a channel protein, also high to low. Water, which is osmosis through aquaporins, which are specific channel proteins for water molecules, that's also facilitated. And then this one, it's a different type of protein, but it's still facilitated. And all of these show what? High to low. So the correct answer would be that they all do. They are all what we call passive transport.
Okay,
Okay, and I'm going to cheat and I'm going to say TX for transport. Okay, just so I don't have to write it out. Okay, and this is the last one. Back to thinking about lipids. Which of these is the correct association? So do trunk lipids have high fluidity? Or do trunk lipids have unsaturated fatty acids? So you have to think about this and tie it together. So what gives high fluidity? So if we want to be high fluidity, then we want to have lots of kinks, okay?
unsaturated.
And so then we want to be unsaturated. And I'm writing like at a diagonal right now. This is lovely. And if we want to have low fluidity, then we want to be unkinked. And we want to be saturated. Okay, so which of these scenarios matches? So what do we want here in the legs, and what do we want here in the trunk or the body? So for the legs that are more exposed to the ice and snow and less insulated, we want the membranes not to freeze solid, right? So that means we want
Saturated.
no, you want high fluidity, right? And here we want lower fluidity because we've got better insulation and we can keep the membrane from freezing with that body heat. So looking at leg lipids, low fatty acid fluidity, nope. Okay, leg lipids, unsaturated fatty acids, okay, low, goes with unkinked, but does not go with unsaturated. So does this one work? Nope. Okay, trunk lipids, unsaturated fatty acids. So trunk, we want low, unkinked, saturated. Does that work? Nope. So what about trunk? We want high fluidity, or did I just mess this up?
We want
Okay, I got to think about this. If we want high, we want kinked and unsaturated. Okay, leg lipids. Oh, it's because of this one. Okay, so this is for sure wrong. This is for sure wrong. Okay, trunk lipids, we want low fluidity, and we wanted them to be
high in fat.
we want them to be unkinked and saturated. So this doesn't work. And I messed up at the very beginning. Okay, let's come back to this one. Leg lipids, unsaturated fatty acids. So we want high, kinked, unsaturated. I don't know why oh, it's because I went with unkinked instead of unsaturated. Does that make sense? So it gets kind of tricky there. So this one, this is what's correct. I totally made a boo-boo there. And now it's going to be on YouTube.
Elephant.
I thought the saturated fats are the ones that were for cold.
No, saturated means that they have all the hydrogen. And so then there are no double bonds with carbon, and they're unkinked, and they pack together. And we want that for heat, because heat makes the membrane more fluid. And so if you are unkinked and saturated, then you are better able to keep the membrane together at high temperatures. And if you if you need to maintain fluidity when it's cold, then you want to push the membrane further apart so that it doesn't freeze and shatter.
Oh.
And so this works. This is where we want things to be cold if we're fighting cold temperature, and this is if we're fighting hot temperatures, okay? Now, I want you to think for a second about what that would mean for cholesterol, because there was another thing that's not shown here. Cholesterol helps hold the membrane together, and it helps to increase fluidity, and it helps to also keep the membrane together. So actually, at both equator and the poles, we would expect to see more cholesterol. Let's see if I can manage to write cholesterol in an even partway legible fashion. We would want more cholesterol in the membranes to both increase fluidity and to moderate fluidity. It kind of works both ways, sort of like a buffer. You would see more cholesterol in organisms that live at the equators and the poles than you would see at places that are mid-latitude, like Washington, or other things that are intermediate. So just to kind of add a little bit on there. Okay, so that is the end of my horrible drawing on this part. Keep my ink annotations. So I'm gonna save this as something else here. I'm still sharing. Yeah, it's lovely. And what I want to do is, when I make this bigger, can you see it? And maybe I'll just do one at a time.
Okay.
So more drawing right here. And I'm gonna, okay, recording yet again. All right, so these are in last week's folder or module. And they say what they are, the 3 and 4 blank diagrams. Okay, so look at that. It is going to let me do that. Okay, so looking at this diagram, which was on your one of your learning assignments, which of the following letters matches the components of the cell's membrane with their letters in the image? So let's just take a look and think about it. Is A a peripheral protein for communication? Well, maybe we should go through and label them. Oh, it's not going to let me write on that, is it? It'll let me. I wonder if I have to do it every time. That's going to be lame. All right. Okay. So this right here, that's cholesterol. So E doesn't match, okay? This one right, oh, let me choose it again. This one right here, A, that is outside in the extracellular matrix, and it's a collagen fiber. And then B right here, if you remember, I identified this last week. So B is a glycolipid for cell identification. And then D right here is a peripheral protein that would help attach the cytoskeleton. And then I've kind of gone over this here, but C is part of the cytoskeleton. And so the only one that correctly matches the letter right now is this one. They're all correct terms, but you would need to move them around to identify them. So A right here matches what this one right here, okay? So this is actually, I know what we can do right now. Should we move them around?
Yeah, I just crossed out the letters.
Yep, okay. So that's A, right? The extracellular matrix collagen fiber. And this is because this was a multiple choice, and so I asked a question here about it. And B is a glycolipid for cell identification. And C, which has now been bumped down, is a component of the cytoskeleton. And then D is a peripheral protein, and E is cholesterol. So now they all match up. Okay. All right, let's look over here. And if I was going to label this guy, these right here, I would label as tight junctions, and I would explain that they are for creating a waterproof barrier. Okay. And these guys right here are desmosomes. So you see them, those are for bolting or anchoring neighboring cells together. And then this one right here is a gap junction, and that's for cell communication. So if I was going to label this, I would put tight junction right here. And then I would call this a desmosome. And I would call this guy a gap junction. Good? And what would you want to write down for each of these? A description of what? What it is that they do.
Waterproof.
Yep.
Yeah.
So waterproof, barrier, and I just lost. Heh, it's in there twice. Okay. Desmosome, we're going to anchor neighboring cells.
Is that for animals
Yes.
only?
Yeah. Well, we have plant cells have cell walls, and they're not really creating. They don't have tight junctions to create tubes like that. They have different cell types that create channels, like xylem and phloem cells, that are very specific. And so, yeah, these are specific to animal cells. And then the
Okay.
gap junction is for animal cell communication. And the one that was for communication for plant cells, those were the plasmodesmata. And there's a picture of that also in the PowerPoint on chapter 3. This was in the first PowerPoint. Okay, so if we wanted to label these guys, would you be able to do that? And so this is just trying to remember. If I pop this open. See this guy? Could you use it to label that? Okay, so, and then could you describe what they're used for? So, let's grab another text box. Oh, that's not gonna work.
Grab your reader's attention. The hook.
Yes, I know, but it's not gonna let me put it over right now. So maybe we'll get tricky again.
Where did the text box come from?
Insert,
Mhm.
and then for me, it's over here on the right, text box.
Oh,
And then you choose a size or a type. Yeah.
thank you.
I'm gonna make this a little bit smaller and split my screen here. Okay, so, and then I think I've lost my scribbling ability here. Okay, so this right here, no, it's not gonna let me do it very well. Let me try this again. Okay, so this is the extracellular matrix out here where it's got the fluid and the things embedded in it. And these things that look all feathery, those are collagen fibers, okay? And then this sticking out through here would be a receptor protein where it's got the specific glycolipid for cell identification. And then what do you call all these little golden bead heads right here?
The phospholipid heads?
Yeah, those are the phospholipid heads. So we want to label these as the phospholipid heads. And then this guy right here that has a channel, that's going to be a channel protein so that things can flow in and out of the cell. Okay. And then I would call this the phospholipid bilayer, right, of the plasma membrane. And then, oh, lost it. This guy right here doesn't look quite the same in this diagram, but that's cholesterol. So you're seeing how it can help maintain fluidity by pushing the phospholipids apart. And the way that it helps to keep the membrane from being not too fluid at the equator was that it has hydrophobic interactions with the neighboring fatty acid chains, and so it helps hold them together. The next thing that we're going to look at here is, do you see this spot right here in this protein? So this is going to be a signaling protein where a receptor molecule could come bind right here and cause a chain reaction inside the cell, because what we have attached to that guy, this
Is
one,
that the active site?
so it would be the receptor binding site. I'm not going to call it the active site. No, it's a binding site for a receptor molecule. Where then this protein changes shape, and in so doing, it activates this peripheral protein. And what's that peripheral protein attached to? We're looking at different filaments of the cytoskeleton here that I just circled that are anchoring the plasma membrane by being attached to proteins, and then they're also facilitating the cell to be able to react when you've got this, binding molecule that attaches, and it can send a signal down through. So the cytoskeleton is doing two things. It's helping with communication, and it's also helping with holding the cell together by being anchored to different proteins. This here, when we see a protein that's all the way through the phospholipid bilayer, we can call it an integrin, I-N-T-E-G-R-I-N. And so we could call all of these integrins, but if we look at this one, it's basically all the way through. It's not serving any purpose for communication right here. Its job here is to basically help provide external structure, because it's got collagen fibers that are going out into the matrix that basically help keep the cell and its extracellular matrix together.
That's a big number.
Okay.
That's kind of weird. Is that a common thing?
A common thing to have. So, red blood cells travel through a fluid, but how does a cell that's part of the heart tissue stay where it's supposed to be? In part, it's because of the interactions that it has with the fluid that it's surrounded by, and in part, it's because it's bound to all of its neighbors. Using the desmosomes and or tight junctions. And so these integrins help maintain, the integrity of the cell and keep it where it's supposed to be in a particular tissue, so that it doesn't just, float away. Okay, So now let's look at this one on the right. This is taking us back in time a little bit, but I wanted to review here. So we're talking about this being the nucleus. Ooh, that was really badly spelled.
Getting a haircut, and I fell asleep.
Okay.
I was like, I almost like my head.
So this is the nucleus, right? And inside,
What time was it?
we have the DNA, okay? And then when the DNA copies itself,
I'm really
it
sick.
makes
Oh, look how fast I'm going.
an expendable copy called the RNA. And then the RNA can leave the nucleus, which is what you see. And the RNA gets read by a ribosomal complex, and we make protein. So I just wanted you to be thinking about this sequence here of DNA, which is in the nucleus, and it stays there. To make RNA, which leaves the nucleus, to then making a polypeptide, which is going to, eventually become a protein, and that's either in the cytoplasm or on the surface of the rough ER. So that's what I was reviewing here. Okay, just that whole when and where, to help you be ready for the next chapter that we're going to cover, and to think about how what's going on in the nucleus of a cell is tied to everything that's going on out in the cytoplasm. Okay. All right, this guy, I'm not going to walk through and type in all of these. This is in the PowerPoint that I had open. This one right here. So the one on chapter 3, that's cell structure. It's slide 31. Okay, if you want to test yourself and look it up, it's slide 31. And I will just kind of talk you through. Okay, so here's the nucleus, and it's contiguous. It shares membrane and passage with the rough ER. And so we can have RNA made in the nucleus that actually goes into the rough ER and gets read in the rough ER and makes polypeptides. But those polypeptides aren't finished proteins that are ready to go, so they have to get processed. And so they get packaged into vesicles, little spheres that are created by budding off of membrane from the rough ER, and they get sent to the Golgi, and there's a receiving face or a cis face on the Golgi, and then the vesicles merge, and inside the layers or the stacks of the Golgi, there are different proteins whose job it is to process the polypeptides that aren't finished. And so they process them, and each layer has a different job. And when they reach the other side, which is called the trans phase, it's basically the shipping phase. And those finished proteins get packaged basically with a zip code that says where they're going to go. And then they get sent. And often when they go through this whole process, when proteins are made in the rough ER and finished in the Golgi and then leave in a vesicle, that vesicle gets sent to the plasma membrane, and those proteins either become a part of the plasma membrane, which tells you how proteins get in the plasma membrane in the first place, or they get released as signaling molecules sent out of the cell. So what's key about this process is we're talking about the endomembrane system. The nucleus connected with the endoplasmic reticulum, which communicates with the Golgi with vesicles. And then the processing of the polypeptide, and then vesicles are created to send proteins to become a part of the plasma membrane or to be released in cell-to-cell communication. And what do all of these structures have in common? What do they share? Membrane. The membrane's contiguous or it's connected by transport vesicles. And so how do we get the plasma membrane of a cell to grow? It's produced where? It's produced in the smooth ER, and it gets shipped this way. How do we get proteins embedded in the plasma membrane? For communication, for passive transport, for receptor molecules to bind to. Those all get processed and made in the rough ER, and then made ready for their full job in the Golgi, and then sent to where they belong in the plasma membrane. Does that help to kind of explain that?
Yes.
Okay. All right, then for this one, it's just, if you saw this diagram on the test, would you know what this little gold head was?
Always.
So the whole thing in the red box
The phospholipid.
is the phospholipid. So the whole thing is a phospholipid, right? And then if we're looking at the head just by itself, that's the polar region. And so it's going to face the extracellular matrix and the cellular fluid, right? And then what would you label the interior as? The little prongs?
Non-polar tails.
Those are the non-polar fatty acid tails. Great. And what is their function? Well, what creates the isolation of the plasma membrane? It is these non-polar tails that all point towards each other. That is what enables the cell to isolate its interior from the exterior.
Jacob, what was the video?
Let me move that out of the way and see if I can get it back. Okay. So
Oh, you were saying.
let me see if it'll let me drag it over up on the top here. There we go. Okay. So basically the fatty acid tails isolate and separate the inside of the cell from the outside. Okay, that's what creates a cell. And then the phospholipid heads are polar and can interact. Okay. There we go. All right. Do we need to do this one again, or you feel pretty good? We labeled this guy pretty well, I think. So I would challenge you to practice this one on your own and see if you can label the different types of passive transport and explain why this one is simple, why these are all considered facilitated, and explain why they're all passive in that we're going from high to low concentration. Okay. All right. I dropped this in here just as a review for you. It was someplace to put it when I found it after the fact that explains what happens to blood cells in different concentrations of salt And so there's nothing to label. I just dropped it in to show you what's going on. Okay. You feeling pretty maxed out? It's almost 9:30 too. We've been at this for a while. What we need to come back and do then on Thursday is cover more with metabolism, and that might be pretty good because you'll be further into it than you are today, having just gotten started. And so that would be the rest of this stuff. So let's take 5 more minutes and just talk about these 2 diagrams to wrap up with enzymes, okay? I talked through this last week, and if you remember, this right here is energy, okay? The level of energy. So we've got high energy here, and then we have lower energy down here. And this yellow box, what we would label this, these are the reactants, and the blue box is the products. And if you remember, it takes a certain amount of energy to break the bonds that are held inside the molecules of the reactants in order to rearrange them to create products. And so we call that energy level that has to be surmounted, we call that the activation energy, and it's a barrier to the progress of the reaction. The peak right here that's red is the activation energy without a catalyst, and the blue peak is the activation energy in the presence of a catalyst. So even though this is an exergonic reaction, which means it proceeds with a release of energy, and it would be what we consider spontaneous, it doesn't occur in cells at, cell temperature. And so in order to get a reaction to occur that's going to release energy that the cell can then use to do work, we have to use an enzyme that's going to destabilize the bonds in the molecules of the reactants, and it lowers that activation energy. It reduces the barrier, and so that then this little hill is something that we can get over, okay? So if you were gonna label this on the test and recognize it, I'd want you to know that the y-axis was the energy stored in the molecules. The yellow here is the reactants, and the blue is the products. That both of these peaks represent activation energy, and that red is the activation energy without a catalyst, and that blue is activation energy in the presence of a catalyst. And that basically a catalyst or an enzyme makes it possible for an exergonic reaction to occur. So let me see if I can insert a text box now. And I want to try to bring it to the front. Where did that go? Maybe I just don't want to do that and I want to try typing. So I'll shrink this guy a little bit and see if it will let me type. Okay, so the enzyme as a catalyst lowers the activation energy of an exergonic or spontaneous reaction so that the bonds between the molecules in the reactants are stressed or strained and become unstable. And what that does is it promotes a chemical reaction
Happen quicker.
so that bonds are broken and new products, and it could be a single product or multiple products are formed, okay? And basically, if you read in the PowerPoint, an enzyme can only catalyze an exergonic reaction, one that's demonstrated here, okay? And so then, if you were gonna compare, you would say, hey, how good am I at doing this? And you would pop open the pop this open and you would test your knowledge and see if you got it right, okay? But the labeling process of the blank one helps this to make sense for you, okay? I'm gonna escape now, exit that. And then what's happening here? This unlabeled one is just getting you to try and label. It's the same as this one here. It's just oriented a little bit different. So see if you can practice labeling that and explain what happens when we have, so what does an enzyme do to catalyze a reaction? So here are the substrates or the reactants. And when they match the active site of an enzyme, they're able to bond. And an enzyme is very specific. It won't just capture any substrate. And so they bond, and then the substrates and the active site change shape. And basically that strains the bonds that were holding the molecules within those substrates or reactants together, and they can rearrange and they become something else. And so notice it was two reactants becoming a single product. And when that chemical reaction happens and we have the new product, the enzyme's active site changes shape back to the original, which kicks the product out because it can't bind. And the enzyme is ready to go through the process again. So an enzyme is recycled repeatedly. They don't get used up. And they're able to catalyze a chemical reaction because they basically make bonds that were stable inside these molecules unstable and therefore easy to break, so that we can rearrange those molecules into something else and spit out a different product. And that's basically summarized in, you know, what's going on here. Okay. So that, I can come back and talk about competitive and non-competitive inhibition later if I need to, but basically it's what is there molecularly that you could use to make it so that an enzyme is non-functional. And competitive means that the molecule actually binds to the active site, so the substrate can't bind, so the reaction can occur. And non-competitive is that it binds somewhere else, and so the active site is still available, but its shape is funny, and so the substrate can't bind. And both of these make it so that a reaction can't proceed. So could you think of, like, if you have a headache and you take medication, if the medication binds to, you know, receptor molecules that process pain signals, then you're not processing the pain signals anymore, and it's competitive inhibition. If it would bind somewhere where the active site shape got changed, but it wasn't actually blocked, then it would be non-competitive inhibition, but it would still function, you know, in the end the same way. And so there are different medications and things that we take that work as competitive and non-competitive inhibitors to basically block enzymes from acting. And I actually have an autoimmune disorder called eosinophil esophagitis, where certain foods that I eat can swell my esophagus shut so that I can't even swallow water. Soy is really bad, but there is a medication that I can take either preventatively or to like for a couple of weeks after I have a really bad reaction. That's a proton pump inhibitor. And it's actually really just heartburn medication, but it works on preventing enzymes from doing their job. And so it's enzyme inhibition. So kind of crazy. So what we have left is to talk about all the diagrams for metabolism. So we'll save that for Thursday. And I see a couple of questions here.